Okay, so our last lecture covered the anatomy side of the lymphatic system, and this particular conversation is going to cover kind of what happens when something does invade your body. And so we're going to look more at the cells that are going to protect you and those things that possibly are trying to get into our system to begin with. Now, before we begin this conversation, the important thing to kind of consider is just how much foreign material we come in contact with in a given day. And it's not even something that we really can wrap our brains around because the number of foreign invaders, pathogens, antigens, things like that, that we come in contact with just through our normal everyday activities an astronomical number. Like I said, just something too big for us really to be able to comprehend. And the fact that we come in contact with as much as we do and we don't get sick and we're not sick every single day is an absolutely amazing thing. Because even on the surface of your skin, if you guys were to take a square inch sample of your skin and look at the number of microbes and bacteria and other things that were living on that skin surface of the skin, it's in the hundreds of thousands. And that's just on the surface. That's not what you're inhaling through air. That's not what you're coming in contact with when you grab a door handle or hang onto a railing when you go up and down stairs. And so, constantly getting bombarded with foreign matter, and your body is 24/7 looking for those things that don't belong. And when it comes in contact with something that your body didn't make or your body doesn't recognize, starting that process of then destroying it before it can invade and really cause any big issues. Now, obviously, it's not a perfect system, and from time to time, those foreign invaders will get the better of our immune system. But like I said, when you consider how much we actually come in contact with and how much time of our lives we actually spend sick, our immune systems do an amazing job. So, with this particular packet that you guys are going to work through, if you start with the first slide, it just talks a little bit about your immune response as a whole. And back when we talked about just your typical body cells, there were three main components that make up your typical body cell. Lipid is by far the main component of your cell membrane. That's going to hold all the contents of your cell together. But also with those lipids, you guys have proteins and you have carbohydrates. And the carbohydrates are really going to form these flags that are going to extend off the surface of the cell. And that's what your body is going to use to recognize the cells that you made. Now, other foreign matter will have its own flag, so to speak, on the surface. But because you didn't make them, you won't recognize them. And that's really what's going to alert your body that this is something that's in our system, but it doesn't belong here. And once that happens, there is this response that's going to be set into motion of your body encountering that foreign matter, marking it, identifying exactly what it is, and then determining how it's going to deal with that foreign matter so it can eliminate it from your system. So, kind of that information that we talked about, like I said, that was probably week 3 of first semester, so it's been a long time. But pulling some of that information that we talked about in the beginning of the year. So, if you go on to the next slide, it talks about cell to cell recognition. And those carbohydrate flags are not only going to allow for your body to recognize the things that it made, it also will regulate growth of your organs. So when the carbohydrate flags of neighboring organs come in contact with each other, they know that they've taken up their space and will slow that growth down to mark how much room and space they actually do have for their growth. Those carbohydrate flags are going to make sure also that your white blood cells don't just start destroying all cells, especially if it's something that you made. And that is really when we get into autoimmune, and I kind of wrap things up in our last conversation talking about autoimmune disorders. But just as a quick recap, those autoimmune diseases or disorders are going to be a situation when, for whatever reason, somebody loses the ability to recognize a particular type of cell that they are producing or that should be there. And once that happens, the problem is that the body will work very hard to destroy those cells, and you're breaking down something that you need to function in a particular way or to keep things in good working order. So those autoimmune diseases, and it can hit and target any cell of the body. So I gave the example of cartilage, but really, if your body makes it, there is the potential that your body could also lose the ability to recognize it. And so autoimmune diseases and disorders are very widespread. If you go down to the next slide, it talks about the immune response. And at the start of the immune response, one thing that I want to do very quickly is distinguish between antigens and pathogens. Now, with antigens and pathogens, some places or some people will use these two terms in place of each other, and you can't because they are two very different things. Antigens are something that can get into your system from an outside source, but you also produce antigens. We have antigens that are associated with our blood type. And so antigens are not always a bad thing, and you can't lump them into that same category as a pathogen. So an antigen is going to be something associated with a cell, and it helps with the recognition. So if it's an antigen that your body produced, again, you should be able to recognize it. But if it's an antigen that is coming from an outside source, you might not be able to recognize that. Now, with an antigen, the thing to keep in mind is that antigens are defined as being something that has the ability to trigger an immune response. Not that it has to, but it has the ability to cause an immune response within your system. This explains why some people are allergic to peanut butter and other people have no reaction to it, why some people are allergic to penicillin and others aren't. While some people can go outside in the springtime and are just fine, and other people start having the runny eyes and the runny nose and everything associated with allergies. Because the antigens that are associated with that pollen that they are inhaling, or the antigens that are found in that peanut butter, when that's introduced to that person's body, it triggers an immune response. So antigens are something that, like I said, it depends. They don't have to trigger an immune response. It does vary from one person to the next. Pathogens, on the other hand, are going to be something that when they invade the body, they are kind of looking to cause destruction or problems. They are invading the source because they are trying to take over our cells so that they can grow and develop in number. So there is a difference between antigens and pathogens. Not all antigens are bad. You guys have antigens that you naturally produce. You will be introduced to other antigens and won't even know it because your body will not respond. And other times, if it's the wrong type of antigen, you will go into that immune response. The last two cells that are talked about on this particular slide talk about B cells and T cells. B cells and T cells are going to be a particular type of white blood cells that we have in the human body. And they get their letter designation based on where they mature. Now, both your B cells and your T cells are going to start in your bone marrow. So that is where they are produced. Your bone marrow is going to make not only your red blood cells, but it's also going to make your white blood cells. So your blood cells originate within that bone marrow. And then what happens is they will mature to a certain degree. And T cells are actually going to leave the bone marrow once they get to a certain point of development. They will go into the bloodstream, travel to the thymus, and they will finish off their maturation in the thymus. And so that is where that T comes from. The T is a designation for the thymus gland where they finish maturing. B cells, on the other hand, they start in the bone marrow and they will stay in the bones and finish their maturation there. And so that B is designated that they are maturing in the bones. So when we talk about B cells and T cells, they are both types of white blood cells. They both are going to start in the bone marrow, but they are going to finish their development in two different places. B cells stay in the bones. T cells are going to travel on to the thymus gland. So if you go over to the next slide, it talks about the B cells a little bit more specifically. And I already talked about where they get their name. But when we talk about B cells have two very important functions that they do. And B cells are not aggressive cells. Their primary response to an invasion of a foreign matter is that they will find something that doesn't belong in the body and they will mark it. So they attach a little mark or flag to the foreign body and then they let it go. They don't hang on to it, they don't do anything to destroy it. They simply mark it or tag it so that as that cell continues to circulate through the body, it's kind of like a neon flashing light to all the other cells of your body that this does not belong here. It's not something that we made and it needs to be investigated further. But the B cells, like I said, once they find them, they're not going to do anything to take them apart, to destroy them, anything in that fashion. The B cells are also going to be responsible for starting the production of antibodies. Now, antibodies are going to be those structures that are going to latch onto a foreign body and kind of hang on to it. And the important part about an antibody is when you have things like a bacterial infection or you have a virus that has invaded your system, they're looking to get into your cells and kind of take them over. The antibody, by grabbing onto them, changes the size of the bacteria or changes the size of the virus. So now you have these incredibly tiny little structures, bacteria or a virus, and you have this antibody that has latched onto it and made it a lot larger than what it typically is. Now that bacteria can't get into your cell because it's too big, or the virus can't get into your cell because it's too big. And so that's one of the ways that antibodies will kind of block and prevent a bacterial or a viral infection, because it eliminates the possibility that those two things can make their way into your cell and take them over. If you go over to the next slide that talks about antibodies, antibodies are different structures. They all have the same base. They look like a Y. So if you think about what a Y looks like, you have the two arms at the top and then you have the one tail coming down. That is the base for all of your antibodies, but there are different classes of antibodies. And the reason that we have different types of antibodies is because they go after different things. And so depending on what is invading your body, the right class of antibody will be released and it will grab onto the foreign material. Now, if you look at the last point, it talks about the antibody. It can't destroy the foreign matter. So whatever it's grabbing onto, it can't do anything as far as dismantling it or taking it apart, but it again is going to prevent it from getting into your cells, and it also just brings more attention to that foreign matter, to your defending cells, your other white blood cells, that it doesn't belong and it needs to be dealt with. One last part on the antibodies before I get too far away from it. One of the things that the antibody will also do is, earlier I talked about antigens kind of being these flag markers, and we have antigens on our own cells, which allows us to be able to recognize that it's something that we made. Antibodies will do something that is called antigen presentation. And so when they grab onto a bacteria or when they grab onto a virus, what they'll actually do is they will take those antigens that are on that bacteria or on the virus, and they will break them off. And those two arms that I talked about, making the Y, they will put the antigen from the bacteria or from the virus on those two arms, and it's called antigen presentation. So now as that antibody is circulating around the body, basically has prevented that foreign material from getting into your cells, but by taking those antigens and attaching them to the arms of the antibody, now as it's floating around, it's telling those T cells or your white blood cells exactly what they're looking for. So that process is called antigen presentation. From there, we have the T cells. The T cells are going to be the aggressive cells that we have as far as taking care of our body and defending it. So as I mentioned earlier, still staying with what are known as lymphocytes. and talked about where they come from and their naming. When they are sent to the thymus, that is where their training is really going to take place. So once they migrate to the thymus, they are going to be trained on how to deal with a particular infection. So you have a group of T cells that are trained on how to fight off anything that you've ever come in contact with throughout your life. So you have a group that is trained to deal with the chicken pox. You guys have had your vaccinations for mumps, measles, rubella, all of those things, polio, smallpox. So you have this group of T cells that is going to be designed to fight off each one of those different types of infections. Now, what that does is that allows you to be very specific the next time that that foreign matter would actually come into your body. So if you guys ever come in contact with polio, you have those T cells, they are immediately going into action. You don't have to build them, you don't have to train them, they are all ready to go. And so as soon as that invader hits your system, you have all of these cells that are going to be ready to attack it and get rid of it before it has a chance to get into your system, take over your cells, and grow in number. And you guys probably would never even know that you had come in contact with it again, because the process of dealing with it would be so swift that you wouldn't even have a chance to really feel bad. You wouldn't start to run a fever or have any of the symptoms that would typically be associated with that illness. If you go to the next slide, it talks about some different types of T cells. Defending the body is very much a process. And in order to be efficient, you are going to have T cells that do different things. And if you guys think about it, you have doctors that have specialties. And that is so when they see a patient, they know their area of medicine very, very well. And they are able to take care of their patients much better because of how specific they are in their particular discipline. Your T cells are very much the same way. Different types of T cells so that they can be very efficient in helping your body. Now, the first class is called helper T cells, and helper T cells are simply going to release a hormone into the body that is going to alert your killer T cells that you have an invasion and that they need to go to work. Killer T cells are exactly what they sound like. Killer T cells are going to be those that when they find the foreign body, they are going to kill it and get rid of it so that it can't do any damage or harm to the body. And in parentheses there, it talks about the fact that these are the cells that you have to worry about when you have somebody that has received an organ transplant, because if you are taking an organ from a different person, they will have their own antigens, potential, even though it's a human organ, that you are not going to recognize these people's antigens, and your body might attack it and try to get rid of it. That's why they do the immunosuppression after you've had an organ transplant, so that it reduces the chance of your body attacking that donor organ. The third class that we have are the memory T cells. Now, memory T cells are very important because these are the ones that are going to find the foreign invaders, and they will learn everything that they can about those foreign invaders so that your body can figure out a way to disarm it, dismantle it, destroy it before it becomes a problem. So the helper T cells alert that there is an invader. Killer T cells are going to eliminate it. And the memory T cells are going to learn everything that they can about that invader so that you can design these cells that are going to be very effective in actually destroying whatever is invading your system. If you go to the next one, this is just a quick comparison between bacteria and viruses. And bacteria and viruses are somewhat different. The first one talks about treatment. Now, if you have a bacterial infection, you can go to the doctors and they can give you a prescription or they can give you an antibiotic and send you on your way, and that will help you recover from that infection. If you are already sick with a viral infection and you go to the doctor, they are going to tell you it's going to have to run its course. Now, a Viral infection can be dealt with a vaccination, but that has to happen prior to you actually getting sick from it. So when we talk about treatment, bacteria, you can treat with an antibiotic. Viruses, if you are sick, at that point, it's going to have to run its course through your body. Part of that actually comes from that cell membrane that you have around the bacteria. The antibiotics typically are going to target that cell membrane, and they are going to break down that cell membrane. That's what they target. They destroy the cell membrane, and once they do that, obviously there's nothing holding the contents together, and it can no longer properly function. Viruses are a little bit different. Viruses are not cells, so they have a different coating around them, and because of that, it's a different process of dismantling it, and the material inside the virus works a little bit different. The number of cells that we have, when you look at a bacteria, and this kind of ties into the size, the bacteria and viruses are much different. Your bacteria is going to be the larger of the two, and usually it's a ratio of about 100 to 1. So in the space where you could fit one bacteria, you would be able to fit 100 viruses in that same space. So they are much different in their size. Bacteria, like I said, much larger, and that ratio is about 100 to 1. The next point talks about the nucleus. Nucleus in the bacteria, it typically is going to be what is called a plasmid. Plasmid means that the nucleus in the DNA is held in a circular fashion. DNA in the human is going to have the spiral formation, that double helix. Again, I know it's been a long time since we talked about that, but this is one of the things that you can use to kind of determine differences between the cells and know if you're looking at a bacterial cell. If that DNA has a plasmid or a round shape, indicates that it's a bacteria. Viruses, on the other hand, they work a little bit different. They don't typically have a nucleus. They don't necessarily need one because of how they work. And not all viruses actually have DNA. They will have some form of genetic information. It's either going to be RNA or DNA, but they are much smaller. They don't have the space, and so they usually will carry one or the other. If it is on the more simple side of a virus, it usually just has RNA. If it's a more complex, then that's when you usually start to see the DNA. But it's also going to be spread throughout. It's not confined. So when we talk about DNA in our cells, it's held within that nucleus. That's not the case with a virus. It's not a cell, doesn't have a nucleus. And so that DNA or that RNA will just be randomly spread throughout the virus. Causing disease. Now, bacteria and viruses both have the potential to cause disease. Bacteria also can have a beneficial function within the body. You guys have natural bacteria that live in your intestinal tract, especially in the small intestine, that help with the digestive process. So not all bacteria are bad. The flip side, viruses, usually when they invade, they are looking to invade so that they can take over your cells and grow and increase in number. But because of how viruses go about the invasion of another cell, they actually are starting to harness the virus and get it to kind of work in a little bit different way. So what a virus does is it has no ability to grow on its own. It does not have any of the genetic information that tells it how to multiply. So we talk about mitosis, cell division in human cells. A virus can't do that. If you have a group of bacteria sitting on the tabletop, they can grow and divide in number just sitting on that tabletop. They don't need to have a host. A virus cannot do that. If you have a virus sitting on a door handle or a railing, whatever's sitting there, that's all that there's going to be, because they do not have the information that they need to divide and increase in number. But you run your hand over it and then you put it into your mouth and it gets into your body. What happens is that virus will now target your cells, get into your cells, specifically go into your nucleus, and it steals the section of DNA from you that tells your cells how to divide, plugs that information into itself, and now it has the information on how to divide and increase in number. And at that point, it's now going to become a growing infection. So they work very differently. What they're doing now with viruses is they are putting it into different situations. They're actually putting it into cancer cells, and they are getting that virus to extract the information from those cancer cells on how to divide. And by doing that, they render those cancer cells, they no longer have the ability to increase and grow in number. And so they're actually using them as a way to shrink cancerous tumors and things of that. Still in the very early stages, but it is showing promise in some of the case studies that they've done and some of the work that they've done with them. Living, I already touched on. Bacteria, yes, they are living. Viruses are not. You can refer to bacteria, they are cells. Viruses are not cells. So you have to be careful in that terminology when you talk about them. So common invaders, antigens, again, don't always have to be bad, but they can trigger an immune response. Pathogens always looking to invade and cause illness and destruction. Bacteria, 50-50, some of them are going to be a hazard. Others do have very beneficial effects. Viruses, even though we're training them to do good things, they are typically going to look to invade and cause problems. Cancer is the only one on this list that we haven't talked about yet. And cancer cells within the human body, cancer cells are going to be those cells that have lost the ability to regulate how often they are going through the process of cell division. And so you get this surplus of cells that don't have a purpose and don't have a need in the body. And once they do that, depending on the type of cancer cells that they are, they can start to invade the surrounding tissues and cause all sorts of problems. So cancer cells are also something that your own immune system is looking for on a regular basis. Most people don't like to think about it, but when you're talking about 10 trillion cells, every person has cancer cells in their body. But your immune system is constantly hunting them, finds them, and destroys them before they ever mass produce and turn into something that is problematic. So these are all things that your body, your white blood cells, your immune system is on the hunt for every single day, would be the things on this list. And then the last one talks about the good cells. We've focused on the lymphocytes, the T cells and B cells, spent some time talking about those as well as the antibodies. I want to focus on the ones that are in the middle. So we have the macrophages, the monocytes, the eosinophils, basophils, and the neutrophils. Now, with those cells that we have, these are the different types of white blood cells that you guys have. And these are the defenders that are going to be circulating through your body, looking for those things that don't belong. And again, anytime that you have different types of the same thing, so we had different types of T cells, that's because they had different jobs. Here we have different types of our white blood cells, and that's because they have different jobs. They go after different things. Now, when you're looking at a blood smear, and one of the labs that we would have done is you guys take slides of blood smears, you put them on the microscopes, and you look at them. Your red blood cells will outnumber your white blood cells 600 to 1. So as you're looking at these blood smears, you're going to see tons of red blood cells. And then every so often, hanging out in the middle of all of these red cells, you'll have this purple cell. It's larger than the red blood cells. It's purple, so it's usually easy to distinguish. And those are your white blood cells. Now, once you've found a white blood cell on the blood smear, the next thing is we take a look at it and we try to distinguish exactly what type of white blood cell it is. Now, with your white blood cells, it's going to have a couple of things that you can use to identify them. Number one, your white blood cells are either going to be granulocytes or agranulocytes. Sorry, I'm running out of time. Okay. So with the agranulocytes, those are going to be your white blood cells that don't have any granules in the cytoplasm. Granulocytes, on the other hand, are going to have these little packages of grains or granules, and so you can see them speckled through the cell. So white blood cells are going to be either granulocyte or agranulocyte. A means without, so it tells you without the grains. And then granulocytes, like I said, you can see the speckling appearance through the cell. The other thing that you can use is you can use the shape of the nucleus. The nucleus in your white blood cells is going to be different. Sometimes it has 5 parts, sometimes it has just 2, sometimes it looks like a horseshoe. On one of them, it takes up pretty much the whole cell. And so that is another distinguishing characteristic that you have with your white blood cells. Now, since this was a lab that we were going to do, I'm going to tweak it a little bit. So since we don't have the slides, and what I'm going to ask you guys to do is I will, in the assignment, list the white blood cells that I want you to talk about. For the white blood cells, you will identify whether or not it is a granulocyte or an agranulocyte. You will tell me 2 things about the nucleus that would help you distinguish what type of white blood cell it is. So is it 5 lobes? Is it a horseshoe shape? But 2 traits that really pinpoint what that nucleus looks like and what type of white blood cell it is. And then the third part is you are going to identify exactly what it goes after. Is it going after a virus? Is it going after a parasite? But what is it looking for within your system? So that's going to be your assignment. I will also type that up so that you guys can see that.